For basically all of human history, we've known that kids look like their parents. It's obvious, right? But how? How does that actually work? Well, that question remained one of life's biggest mysteries for thousands of years. Today, we're going to crack that code on a journey that takes us from a quiet monastery garden all the way to the blueprint of our own DNA.
So let's just dive right in with a question for you. Have you ever wondered why you have your mother's eyes, but maybe your father's smile? How does life pass down its instruction manual from one generation to the next with such incredible precision? That's the fundamental puzzle we're going to solve together today.
You know, the quest to figure this out is ancient. For nearly 10,000 years, we've been using selective breeding with crops and animals. We knew traits were passed down. But it wasn't until the 1860s that an Austrian monk, Gregor Mendel, started to find the first real clues. And here's the wild part. His game-changing research paper went completely unnoticed for over 30 years. It was finally rediscovered in 1900, and that's what officially kicked off the science of genetics. So let's meet the hero of our story. In a quiet, unassuming monastery garden, Gregor Mendel was about to crack a code that had stumped pretty much every great thinker before him. A huge part of Mendel's genius wasn't just the questions he asked, but his choice of subject. He picked the simple garden pea. Why? Because it had clear, simple traits and grew really fast. Honestly, this methodical choice was the key that would unlock everything. And here's what was so brilliant about his method. Instead of getting bogged down in complex traits, he focused on 7 simple either-or characteristics. A plant was either tall or dwarf. Its seeds were either round or wrinkled. There was no confusing middle ground. This totally sidestepped the old idea of blending, which had led so many other people astray. So, why did Mendel succeed where everyone else had failed? Well, first, he kept it simple, studying just 1 or 2 traits at a time. Second, as we said, the pea plant was perfect. It grew fast and had tons of these simple traits. But here's the real game changer. For the first time ever, someone applied math and statistics to biology. He didn't just look, he counted. And because he used huge sample sizes, his results had undeniable credibility. Okay, let's walk through his most famous experiment. First, he crossed a pure-breeding tall plant with a pure-breeding dwarf plant. In that next generation, the F1 generation, something really weird happened. All the offspring were tall. The dwarf trait had completely vanished. But then, and this is the good part, he let those tall plants self-pollinate, and in the next generation, poof, the dwarf trait magically reappeared. So where did it go, and how did it come back? This is where all that counting paid off, big time. When he tallied the results from that second generation, he found a stunningly consistent, predictable pattern. For every 1 dwarf plant, there were 3 tall plants. A perfect 3-to-1 ratio, over and over again. This wasn't random. This was a clue. It was a mathematical law hidden right there in nature. This discovery led him to his first huge idea, the law of dominance. He figured that for each trait, an organism has 2 factors. When those factors are different, one just masks the other one. The factor that shows up, like tallness, he called dominant, and the one that gets hidden, like dwarfness, he called recessive. This perfectly explained why all the plants in that first generation were tall. They all had that dominant tall factor. But how did that dwarf trait make a comeback? That's where his second law comes in, the law of segregation. He realized that the 2 factors for each trait don't blend or merge. Instead, they separate, or segregate, when sex cells are formed. So each parent only passes on one of its 2 factors. This meant that the hidden dwarf factor could be passed on, just waiting to reappear if it happened to pair up with another recessive factor. Incredible! Traits were passed on as distinct, separate units. And he didn't stop there. He started looking at 2 traits at once, like seed shape and seed color. And he discovered that inheriting the factor for round seeds had absolutely no effect on inheriting the factor for yellow color. The traits were sorted independently of each other, kind of like shuffling 2 totally separate decks of cards. This independent shuffling created its own predictable pattern and became his third great law, the law of independent assortment. So Mendel had laid down the fundamental rules of the game. But as other scientists started digging deeper, they found out that the story of heredity was actually even more complex and way more fascinating than he ever could have imagined. It turns out not everything is strictly dominant or recessive. With incomplete dominance, the traits actually do kind of blend. A red flower and a white flower can make a pink one. Then there's codominance, where both traits show up, fully and equally. The best example of this is our ABO blood type, where the A and B alleles are codominant, creating the AB blood type.
And get this, sometimes a single gene can be a total multitasker, influencing several different traits that seem completely unrelated. This is called pleiotropy. A classic example is the single gene mutation that causes sickle cell anemia. Amazingly, that very same gene also provides resistance to malaria. One gene, multiple very different effects.
So what about traits that aren't so simple? I mean, things like human height or skin color show this whole beautiful range of variation. That's because they aren't controlled by just one gene. They're controlled by multiple genes, all working together in what we call polygenic inheritance, where the effect of each gene sort of adds up.
All of this was incredible, but it raised a huge question. Mendel talked about these abstract factors, right? But where were they? What were they actually made of? The hunt was on to find the physical evidence of heredity inside our very own cells. The breakthrough came in 1902. As scientists were peering into their microscopes, watching cells divide, two researchers, Walter Sutton and Theodore Boveri, noticed something amazing. The way these little structures called chromosomes behaved during meiosis, it looked strikingly familiar. In fact, it was a perfect mirror of what Mendel's factors were doing. I mean, the parallels were just undeniable. Mendel's factors came in pairs, so did chromosomes. Factors segregated when gametes were formed, chromosomes did the exact same thing during meiosis. Independent factors assorted independently, so did independent chromosomes. It all clicked into place. They proposed the chromosomal theory of inheritance. Mendel's factors, or as we call them, genes, must be located on chromosomes.
Now, a theory is great, but you need proof. You need that smoking gun. And that proof came from T.H. Morgan and his legendary work with tiny fruit flies. His experiments not only proved that genes were on chromosomes, but also led to another huge discovery called linkage, the idea that genes sitting on the same chromosome tend to be inherited together as a package deal.
So, from peas to fruit flies, the core principles were locked in. Now it was finally time to apply all this knowledge to ourselves, to start decoding the human blueprint, and to truly understand our own health and diseases. And this is where all this science becomes incredibly personal. Remember how chromosomes are supposed to segregate? Well, sometimes errors happen. Down syndrome, for example, is caused by having an extra copy of chromosome 21, a condition known as trisomy 21. That single change to the blueprint has massive effects on a person's development. And these errors can also involve the sex chromosomes. Klinefelter syndrome happens in males who have an extra X chromosome, making their genetic makeup 47XXY. This typically leads to sterility and the development of some feminine characteristics. On the flip side, Turner syndrome is caused by the absence of one X chromosome in females, giving them a 45XO karyotype. This also results in sterility and underdeveloped secondary sexual features. All of these conditions are powerful real-world examples of just how critical a precise number of chromosomes is for our development. Just think about how far we've come, from a monk just noticing patterns in his pea plants to our ability now to read our entire genetic sequence. We've unlocked the blueprint of life itself. Our genetic code is the story of our past written into every single one of our cells. And that leaves us with one profound question. How will we use this incredible knowledge to write our future?